but Jesse will walk you to the next.
Okay. Is it you would pick people up for this, or
I have a recitation I have to teach.
a little bit?
Oh, for you,
For me?
yeah. The grad lunch will be.
Okay, cool.
Yeah.
Awesome. Wow, it's already lunch.
Yeah.
It's crazy.
All right, it's 10 o'clock, so I guess I will get started with an introduction. So I just want to say hello and thank you for being here. We're very excited to welcome Dr. Chris Funk as our, sorry guys, I'm not to the microphone, so thank you for joining us. And what should I do, Ricardo? Yeah, maybe I'll do that. That's all. Okay. So thank you for being here. We're very excited to welcome Dr. Funk for our Integrative Biology Graduate Student Association's Invited Speaker for 2022. We're really excited to listen to that in person this year. It's the first time that I've gotten to do it in person, so we're really excited about that. So before we get started, we do want to take just a couple of minutes to thank folks who made this event possible. So thank you to the department for funding this event and allowing it to happen. Thank you to the great team in the office, Tracy, Tara, and Teresa. They're amazing and we couldn't have pulled it off without them. Thank you to the Invited Speaker Committee and the Seminar Committee, Ricardo, especially for helping us get this set up. And to all the grad students who participated during the speaker selection process and everyone participating today, your participation is what makes it worthwhile. So thank you all. So before I hop into my introduction, of course, thank you, Dr. Funk, for traveling all the way here to be with us. And we're very excited about you. Into the introduction. Dr. Funk earned his bachelor's degree in biology at Wesleyan University and PhD at the University of Montana, he was a postdoctoral fellow at the University of Texas and completed postdoctoral research at the U.S. Geological Survey here in Corvallis, Oregon. He served as an assistant professor at the College of William and Mary before moving to Colorado State University, where he is currently a professor in the Department of Biology and director of the Colorado State University Global Biodiversity Center. In addition to the many awards and accolades he has received, his research investigates how evolutionary and ecological mechanisms create and maintain biodiversity. His research spans several taxonomic groups, involves the use of population genomics, quantitative field methods, controlled experiments, and computational analysis. His lab seeks to reveal new insights into conservation genomics, disease transmission dynamics, and sensitivity to climate change, and he works with groups nationally and internationally to use these understandings to inform conservation policy decisions. So with that, I would like to welcome Dr. Funk and invite him to get started.
Echoing? Okay, hopefully not. Thank you very much for that very generous introduction. It's I also really want to thank Katie for organizing my visit and the IB graduate students for inviting me. I'm a very proud Oregonian, to the point that apparently it's a little bit annoying, according to my wife and friends. So it means a lot to me in particular to be invited to give a talk at my home state land grant institution, which is hands down the best university in the state, my unbiased opinion. Go Beavs! See if this is going to work out. Awesome. All right, so let me give you a little outline of where I'm going to take you today. So first of all, I want to give you a little sense about my background, why I got into the field, and what I do, because if you look at my website, it's like, what the hell does this guy do? Then I want to give a general introduction to the topic of my talk today, which is trying to understand the importance of interspecific trait variation for predicting vulnerability to climate change. And then we'll dive into a couple of ongoing research projects in my lab. The first will be talking about a genomic approach to try and understand vulnerability to climate change and the genomics of adaptation in Channel Island song sparrows. Then I'm going to move on to a project that's very close to my heart on vulnerability to climate change in the amazing tailed frog, which lives right here in the Pacific Northwest. And this is just some emerging results. I've not presented them yet, so I'm really excited to talk to you about what we're finding so far. And then just finish with some concluding thoughts about what it all means. All right, so first of all, a little bit about my background and research program. And I think this is particularly relevant to being here today. So I feel very lucky to have grown up in the beautiful and ecologically diverse state of Oregon, which I think played a huge role in my love of nature, scientific discovery, and conservation. In fact, I know it did. So I always show this slide in talks that are not in Oregon to explain to people that the entire state is not a rainforest, it's not all wet, and actually a lot of the state is high desert. And this ecological diversity is one of the things that makes Oregon such a special place to be a biologist. So like a lot of people who've gone to biology, I've loved being outdoors as long as I can remember. Oh yeah, one thing I was going to say that's kind of important is that I grew up right here where that star is. And it's a little hill, little bump in the Willamette Valley called Cooper Mountain, just about half hour outside of Portland. And the reason that's important is because probably the reason I got into biology is I spent all my time as a kid outdoors, running around the forest. This is before that, you know, you had to keep track of where your parents are when they're coming home. If I wanted dinner, that I should show up in time for dinner. And then as I became a young adult, I continued to enjoy the outdoors anyway I could, doing things like backpacking. Who's ever been on Camellia Creek Trail up in the Willamette National Forest? Rock climbing. I was even a wildfire fighter for summer. And my favorite outdoor activity was whitewater kayaking because rivers were always my favorite environment. So biology was a natural fit for this night kid who just loved to be outdoors all the time. But then something happened that turned a budding young biologist into a budding young conservation biologist. And so I took these photos when I came home from college one summer, when I found my forests on Cooper Mountain just totally fell and clear cut. You know, I understand at the time those were my forests, but actually they belonged to someone else, and they chose to sell the wood. So this broke my heart and pissed me off. But eventually I got to the point where it also inspired me to try to make a difference. And so this is a common story that probably a lot of you could tell as well. This is also a story that I've heard from the late Michael Soule, who was the father, grandfather of conservation biology, who was motivated to try to conserve wildlands and biodiversity when he saw his favorite arroyos outside San Diego destroyed or developed. And of course, this is exactly what was happening on my beloved Cooper Mountain and Willamette Valley. If you're spending time in Washington County, you see a lot of this, a lot of cookie cutter homes, and this is pretty rampant in Washington County. And though I didn't understand it at the time, what I was witnessing was part of this global trend that has been called the Great Acceleration by the International Geosphere Biosphere Program. And I think this figure does a great job of just giving a sense of the unparalleled rate of change in population growth, per capita consumption, all the things go with it, things like McDonald's restaurants, all right, that are driving the biodiversity crisis. So one really major aspect of the biodiversity crisis is obviously habitat loss and fragmentation, as represented here by slash and burn agriculture. And of course, the big one that's on our mind is climate change, right? We all know that average temperatures have already increased by about 1 degree Celsius since the Industrial Revolution, and they're projected to increase a lot more, even under the most optimistic scenarios, all right? Unfortunately, we're currently headed towards one of the more pessimistic scenarios. And we've seen back-to-back record increases in global temperatures, like 2015, global record, 2017, global record, et cetera. Now, this is not the world record. You want to strive to break. So this all leads to where I am now. What do I do? What's my motivation? And so ultimately, my big picture sort of motivation of what I want to do in research is try to understand the evolutionary ecological mechanisms that generate and maintain biodiversity. And at the same time, I strongly believe that as scientists who study biodiversity, it's our responsibility to share our knowledge with policymakers, conservation practitioners, because they're the ones in the trenches making these difficult decisions. We want them to get the best information possible to make those decisions. And so my happy place, my favorite questions are questions that both address fundamental questions and ecology and evolution, address theoretical questions, but also have direct conservation implications. So my research is really integrative. I combine field research, originally I'm obviously a boots on the ground kind of guy, experiments, got into genomics, basically, well, genetics at the time in college, and then genomics goes hand in hand with computational approaches. This is why my Twitter handle is field genomics, right? So it's a combination of field work and genomics. And then I integrate these approaches to address a diversity of questions related to biodiversity conservation primarily. What I work on, I don't know, I work on a bunch of different things. This happens when you're a geneticist and people are like, oh, I need a geneticist on my project. You want to work on slided owls or map lines or whatever. And so I work on a huge variety of animal species, primarily vertebrates, but also some invertebrates. I just started working on monarch butterflies. And I work all over the Western Hemisphere so far, ranging from northern temperate forests down to the tropics. I spent a fair amount of time in the tropics. I'm not going to talk about that research today, but I've been working down in Ecuador for about 25 years and also Colombia, Peru, et cetera. And it's obvious from the Funk Lab logo, my favorite environments are mountains, streams, other aquatic environments. And if you grew up in the Portland metro area, this mountain looks familiar. I think I didn't intend to make it Mount Hood, but basically it's Mount Hood. And there's a lot of great things about our jobs as professors, right? We get to basically be kids our whole life trying to investigate questions and go out in the field and delete a bunch of emails. But I think the best part of my job is training the next generation of biologists. And so this is just a hodgepodge of some of the, most of the postdocs and graduate students and undergraduate students I've had the pleasure of working with over the years. And Dusty here, I don't think he's here right now, but this guy right here, Dusty again, was an undergraduate in my lab, came to Oregon State and just defended in botany and plant pathology, I think, last week. So that's awesome. I make a special effort to try to recruit diverse people because, ultimately, what is science? We're trying to understand truth through observation and experiment. And that belongs to all humanity, right? And also, I think that these diverse perspectives, people with different backgrounds, come up with different creative solutions to these super challenging, wicked problems about biodiversity conservation. In terms of research subjects, I'd like to think of my research program as a tree. So the trunk and roots of the tree is trying to understand the mechanisms that generate biodiversity and figure out solutions to conserve it. And then I currently have 4 sort of main branches of investigation in my lab. The first of these is conservation and population genomics. Another one is climate change, vulnerability, and resilience. Another angle is adaptive divergence, trying to understand the microevolutionary mechanisms and environmental drivers of adaptive divergence. And then also tropical ecology, evolution, and conservation. And then these leaves can be thought of specific research projects on these branches. And one leaf from the conservation and population genomics area is that I was invited by my former PhD advisor, Fred Allendorf, to help him write the 3rd edition of this conservation genetics text because it was outdated. It didn't have all the new genomics stuff in it. And this is the first book that's fairly comprehensive, talking about how you can use genomics, apply genomics to conservation problems. And I have to say, after working obsessively on this thing for 3 years, my poor family, it was a very good feeling to finally hold it in my hands about a month ago. And actually, I have some show and tell afterwards if anyone wants to take a peek. I was going to hand it around, but I don't want Balloon or Lytle to get distracted during the talk and not be able to concentrate. So welcome to take a look at that later. Another leaf in my research program is understanding mechanisms causing speciation in frogs and mega diverse tropical countries like Ecuador and Colombia with a ridiculous amount of biodiversity. One of my favorite projects right now is trying to understand vulnerability to climate change. You amazing tailed frog, raise your hand if you know about the tailed frog. There's 1, 2, awesome, 3 back there. I'll talk about why it's so amazing. And that's right here in the Pacific Northwest. And then another leaf on the research program tree is trying to understand microgeographic adaptation and songbirds and the California Channel Islands. So this is what I'm going to talk about today in combination with understanding vulnerability to climate change, because I think these two topics are really intrinsically interlinked for kind of strange reasons I'll explain about today. And as Katie mentioned, in addition to research and teaching, I also lead this center at CSU called Global Biodiversity Center. And our goals are to basically promote biodiversity conservation through research, outreach, and teaching. And thanks in part to our efforts, CSU was recently ranked as top 10 in biodiversity conservation by the Center for World University Rankings. All right, so that's kind of what I do. So now that you have a general idea of what the heck I'm doing with my time, I want to dive into science and start with a general introduction to the theory on why interspecific trait variation matters for doing a good job of understanding vulnerability to climate change. So biodiversity is going to take it in the pants with climate change. So this is a meta-analysis that Mark Urban did a few years ago, published in Science. And basically what he's trying to do is look at all these studies that have looked at the effects of climate change on biodiversity as a function of different emission scenarios. And not surprisingly, it's a positive relationship. And what he found is that if we're able to stay within the 2 degrees Celsius sort of target, we're looking at about a 5.2% loss of biodiversity. However, we're more like around here. And so we're looking at more about twice that loss of biodiversity. For some people, hearing conservation values are like, oh, 10% loss of biodiversity, that doesn't sound so bad. But this is not including all the other interacting factors, habitat loss, fragmentation, invasive species, exploited populations, disease, etc. So it's, cumulatively much worse than this. But before we go on, one thing that's clear is, although there's this trend line right there, look at all the scatter. There's tons of variability in these projections. Some of this is real variability among communities and species, but some of this is just uncertainty, because this is tough business to try to understand how things are going to respond to climate change. So one way we can move the bar forward in trying to improve and trying to make a difference in terms of climate change as scientists is to do a better job of predicting vulnerability to climate change. So what is vulnerability? You hear about this all the time, probably on a daily basis in this department. So vulnerability is ultimately sensitivity times exposure minus adaptive capacity. What the heck does that mean? So let's go through each of these terms and define them. So vulnerability can just be thought of as the probability of extinction caused by climate change. So essentially, the y-axis in that urban figure is a measure of vulnerability. Sensitivity is the response of a population of species per unit increase in climate change. It's like a 1 degree Celsius increase in climate and temperature. How is that going to affect your population? A lot of times people conflate sensitivity and vulnerability, but they're different because vulnerability is also a function of exposure. So the actual magnitude of climate change. Obviously, temperature is not the only dimension of climate change, but that's what I'm focusing on here. And so vulnerability is sensitivity times exposure minus adaptive capacity. And it can be modulated by adaptive capacity, which is the ability of the system to adjust to climate change through dispersal, move to a better place, plasticity, or evolutionary potential. And I think it's fairly well recognized that genomics and evolution has a role to play in quantifying and understanding dispersal and gene flow, as well as evolutionary potential by definition. But I'm going to argue today that genomics is also, an evolutionary perspective is also really important for understanding variation and sensitivity among and within species. So focusing for now just on sensitivity, that's what I'm mainly going to be talking about the rest of the day. In order to understand sensitivity, we have to focus on what I call sensitivity traits. So these are traits that mediate the effect of climate change on fitness and population responses. So examples of climate change sensitivity traits could be things like thermal tolerance, which I'll talk about a lot today, drought, flood tolerance, or if you're a marine organism, particularly one with shells, then a critically important sensitivity trait is your tolerance of acidification. But of course, climate change is not the only rapid environmental change going on right now. And there's lots of other radical changes. And so you can imagine things like disease resistance being a very important sensitivity trait, or competitive ability against a basis. You can go on and on. But it's important that you think about traits. The take-home message here is that really pushing the bar and understanding vulnerability is going to require thinking about traits and thinking about sensitivity traits that mediate the effects of climate change on populations. So going one level deeper, you can ask, well, what's determining this variation among species within species in sensitivity traits? We know that some species are more sensitive than others. And so my colleagues and I, as part of a grant proposal, you need to learn how to make conceptual figures, very important skill. We came up with like a very simple conceptual framework for thinking about this problem. It starts out with landscape and environmental heterogeneity that organisms experience in their evolutionary history. So right away, evolution is important. These in turn affect evolutionary processes like selection, genetic drift, and gene flow. And those in turn determine the distribution and amount of genetic variation across the landscape. And then this genetic variation, in combination with direct environmental effects, determines the distribution of traits across the landscape. And right here, I'm talking specifically about sensitivity traits, so traits that mediate the effect of climate change. And then ultimately, this phenotypic variation determines relative vulnerability among populations, different parts of the species range, etc. So this is, I mean, if you're a population geneticist, it's just like your view of the world. And all I did is add sensitivity traits and vulnerability. But I think it's true. So what I want to argue today is that it's really important to understand local adaptation in sensitivity traits in order to predict which populations, which part of the species range are going to be most vulnerable to climate change first. Right, so this panel right here shows a very common mindset in climate change vulnerability assessments where species are considered to be fixed entities, where this, you just say, this is the sensitivity of the species. There's no variation within the species in sensitivity traits. So the way to look at this is that, first off, this blue line is temperature, in this case across elevation, but it could be any temperature gradient that you prefer. This blue dashed line is critical thermal maximum, CT max. And right here, I'm assuming that there's no variation across elevation. And then this orange line shows your future max temperatures. This might be like your September, August temperatures in streams, for example. And if there's no variation in sensitivity traits across elevation, as shown here by this flat dash line, then of course the low elevation populations are the first ones that are going to be impacted by increasing temperatures. But imagine if there is variation in sensitivity traits. If you're an evolutionary biologist, you assume there's local adaptation. So if there's local adaptation and CT max varies as a function of elevation and temperature, then as temperatures increase, then they'll all be potentially equally vulnerable or not vulnerable, depending on how much increase in temperature there is. Or imagine a case where CT max could evolve. And we know, based on a lot of different data in a lot of different systems that oftentimes low elevation populations have more genetic variation than high elevation populations. If that's the case, they might evolve more. And so you might have a case where the future CT max looks like this, where the low elevation ones evolve higher CT max more than the high elevation ones, in which case the high elevation ones could be most vulnerable. So the point here, I mean, you could go on and make all sorts of predictions and hypotheses, but the point here is that understanding how sensitivity traits vary across the landscape is super important for understanding who's going to take in the advance first. So I think two really big important questions that we need to answer to do a better job of predicting vulnerability to climate change are, first of all, how common is interspecific variation in sensitivity traits? And also, maybe more importantly, can we predict the distribution of that variation across the landscape? And also, at what spatial scale can local adaptation and sensitivity traits occur? Is it microgeographic? In other words, does it occur at a really fine scale within the dispersal distance of species? Because if it does, it means there could be a lot of heterogeneity across the landscape in the amount of and the relative vulnerabilities to climate change. All right, so that's a little theory and kind of frames what I'm going to talk about with empirical results today. And the first thing I want to talk about is the genomics of thermal adaptation in Channel Island songsters. So this is research that was led and is actually still being led by Dr. Madeline Gambeau, who defended last year. And she's currently a visiting assistant professor at Colorado College. She wants to go more of a teaching track, which is awesome. And she was co-advised by myself and Cameron Gamble at CSU, also Scott Stilt with the Smithsonian Migratory Bird Center, and Nature Conservancy. Scott Morrison was not an advisor, but a really important collaborator in this project. All right, so Channel Island songsters, very interesting subspecies. So historically, these guys are found on 6 of the 8 California Channel Islands right off the coast of Southern California. So Los Angeles is this big giant monster over here where the song sparrow is on hiding Los Angeles. And historically, it was found on these 6 islands, but has since gone extinct on this little island here called Santa Barbara Island, and this island here called San Clemente Island. Fortunately, the Park Service, which owns and manages this little rock right here, Santa Barbara Island, wants to reintroduce the song sparrow, which is a pretty exciting opportunity for us to try to help them do a good job. So before the ranching era, which occurred 1800s up and through most of the 1900s, the Santa Barbara Island had a breeding population of song sparrows. But then, for people so they could bring things we like with us, we brought cats, we brought rabbits. Actually, this was not taken on Santa Barbara Island. Guess where I took this, which is even sadder. San Cristobal Island in the Galapagos Islands, right? They have just all sorts of feral cats all over the place eating these endemic gill warblers. Anyway, so they introduced these cats and rabbits out here and then found that there was a fire sucker punch, and then whoop, there go the song sparrows. But since then, the Park Service has removed the cats and the rabbits and did a really intensive job trying to revegetate the island. So we got an islandless vacant, introduced mammals, and also song sparrows. So it's ripe for the reintroduction of these song sparrows. But importantly, one thing that should be considered in deciding where are they going to get these guys, well, I should mention where are potential sources. There's several potential sources. So the subspecies is found on Miguel Island, Rosa, Cruz. They could get birds from any of those islands or more than one island if they wanted, just throw them all on, let natural selection work it out. Or they could take them from the mainland, but the mainland is actually a different subspecies, so maybe they don't want to do that. But one thing they should consider in making this critical choice of where they're going to get the birds from is a really pronounced climatic gradient across the islands. All right, so San Miguel Island is hit by the California current, so it's super foggy, super windy, pretty cold, versus Santa Cruz is relatively shielded from the California current, and it's relatively hot, not usually as windy or as foggy. And we like to say that going from San Miguel Island to Cruz is like going from Scotland to Baja California over this really small climatic gradient. And importantly, the late Russ Greenberg, a great ornithologist who worked at Smithsonian, passed away several years ago. He did some cool work looking at museum specimens, and he found that residual bill surface area, so this is bill surface area corrected for body size, is positively correlated with the average maximum high temperatures, right? And this is specifically looking at the Channel Island song sparrow. And they hypothesized that this is because bill surface area is acting as a radiator. So bills are highly vascularized, as shown in this micro CT scan, which allows them to lose dry heat without losing a lot of moisture. That's pretty cool. And you can also see in this thermal image that the bill temperature is quite a bit warmer than the core body temperature. So it's thought that this is acting as radiator. On the hot island, you should have a bigger bill and lose heat more effectively. But this is just a hypothesis. It's a very opportunistic collection. And so certainly there's no conclusive evidence that adaptation has occurred across these islands. And so we went back and tried to dive into this question more deeply. So our overarching question here is, are song sparrows on different islands adapted to different climatic conditions? This is a big question, so we broke it down into some smaller bite-sized chunks. First off, we want to go back and do a more intensive job at looking at the pattern. So do contemporary island populations differ in this thermal regulatory trait? Right here, we're focusing on the bill morphology, but Maybelline did do a lot of other physiological work as well that I'm not going to go into today. Second question is, how much gene flow occurs between islands, and what's the relative importance of genetic drift? Are these populations the traits just drifting because of small effective population size? We got this using neutral SNP or single nucleotide polymorphism loci. Third question is there evidence for divergent selection among ions? We got this using a couple of different approaches for testing for divergent selection. I'll get into those later. And lastly, are there loci associated with bill variation? And we've got this using a genome-wide association study, because all this variation could just be driven by phenotypic plasticity. We don't have any evidence yet that this is genetically based, and therefore that it represents evolution. And so ultimately what we're trying to do is look at doing the best we can with the non-model species to infer adaptation. And so in the case of the song sparrow, we're looking at phenotype environment relationships. The phenotype is bill morphology and primarily focused on temperature. Then we're using landscape genomic approaches to test whether there's any SNPs that show a signal of divergent selection that's driven by climate. And then lastly, we're looking for relationships between bill morphology and specific variants to test whether it's genetically based. All right, so first question, do contemporary island populations differ in these thermoregulatory traits? As you can see by the distribution of these sampling points, Maybelline is amazing in the field. So like, these may look like nothing on the map, but seriously, like a Miguel, you get dropped off, there's one little office, there's no roads, it's, the wind's like 50 miles per hour, and Maybelline went and figured out how to misnet song sparrows in 50 mile per hour winds. She carried like a big volleyball pole with her with a big concrete block. So it's really intense field work. She also got all over Santa Rosa Island, once again, super intensive, and cruise. And then when she caught birds, she did blood sample, and then she'd do a thorough job of measuring bill dimensions as well as tarsus length, so we could quantify this trait. So prediction here, based on the theory and also Russ Greenberg's work, is that San Miguel birds will have relatively proportionally small bills because it's cold, you don't want to lose heat, versus Santa Cruz, it's relatively hot, you have relatively large surface area, so you can lose heat. And then Rosa is in the middle. So what did we find? Well, she found exactly this pattern. And there's a lot of data points there because she collected a lot of birds, as you can see. And even though there's so many other factors that can affect build dimensions, and there's going to be some error in the measurements, the R-squared value was 0.27, which is pretty big for biology and evolution in my book. And so this is pretty exciting, but once again, by no means demonstrates local adaptation because we don't know if there's divergent selection, we don't know if it's genetically based, et cetera, but the pattern is there overwhelmingly. Importantly, you're thinking, well, what about Darwin's finches? Maybe there's different diet. There's one right there. It's Darwin's diet. So another hypothesis is that there's different food on the different islands, which is driving divergent selection on foraging ability, so they feed on different things more efficiently. And so Maybelline also tested this as rigorously as she tested everything else. And she looked at bite force, she looked at seed extraction time, and she looked at food resources among regions. And she found no evidence of variation in bite force or seed extraction time related to bill size. And the food resources differ a little bit, but not in a pattern that would explain this variation we're seeing. So this is not another Darwin's finch story. So the second sub-question is here is how much gene flow occurs per island, and what's the relative importance of genetic drift? So what's the effective population size? If you have a small effective population size, it's lots of drift. Larger effective population sizes, not so much. So to get this question, Maybelline generated a Grad data set, about 22,000 SNPs, and these were all neutral loci. She got rid of anything that showed any signature selection, and then she inferred gene flow and estimated effective population sizes. So here's the distribution over sampling. It's 450 birds. This is a pretty big data set for most genomic studies, unless you're working on specific cell lines, and then it's like 450,000. And what you found is that genetic differentiation was pretty high between the mainland birds and the islands. For birds, these are pretty high FST values. So FST of 0 means there's no difference. FST of 1 means they're fixed for different alleles. And this is relatively high for birds. If you look just among the islands, you get lower FSTs, which probably is not surprising. But these are still pretty substantial FSTs for birds over this kind of spatial scale. For birds, this is a microgeographic scale. If you're working on salamanders, they have FST values like this, 10 meters apart underneath different blocks. She also used a clustering algorithm, and she found really strong support for distinct genetic groups. There's a distinct San Miguel group, Santa Rosa group, Santa Cruz plus Anacapa group. What's interesting about Anacapa is when we started the study, there weren't song sparrows out there, and then a couple years later, they had flown out. So these probably just represent recent colonizers from Cruz. This is due to removing like rats and mice out there so that they could establish again. All right, so another thing that's kind of interesting to note, you probably can't see it from back there, but there's actually a red line right here in San Miguel. So this is a Cruz bird that has gone over to Miguel against the wind. And there's also a Santa Rosa bird that does family Santa Rosa that maps back genetically to Cruz. So there is some ongoing dispersal. And this shows like, these are the kind of sample sizes you need to be able to detect contemporary dispersal using basically assignment tests. Also, she found pretty large effective population sizes out there. So the smallest was Santa Cruz Island, which is NE of 500. This is a big effective population size compared to most of the critters I work on. Rosa was 1,300. For comparison, I've worked on the population genomics of island foxes on these same islands that have gone through bottlenecks and stuff. And in that case, the NEs are all under 20. Where in that case, genetic drift is the overwhelming most important microevolutionary factor. But this suggests, okay, you've got relatively low gene flow per bird, and you've got, well, let me just kind of review. We know that birds in hotter areas have larger bills, there's relatively limited gene flow, and relatively large effective population sizes. So low gene flow, large effective population sizes are the perfect conditions for selection to be effective at causing changes in allele frequency, so actually causing adaptive evolution. So then our next two sub-questions are, is there genomic evidence for divergent selection and loci-associative morphology? And so the third question here is there evidence for divergent selection among the ions? And to get at this question, there's a couple standard approaches for testing for loci under selection. You can do FST outlier tests where you're trying to find loci that have much higher divergence than the background level, the background neutral rate. Or you can do these genotype environment association tests where you're looking for these loci that have this really pronounced association with an environmental gradient. And what she found is, sure enough, using both approaches, there's pretty strong evidence for many loci that are under divergent selection. So a total of 41 loci based on the FST outlier test, 34 based on the GA test, and 4 overlapped. And importantly, when you do the gene annotation, some of these genes make sense in terms of the trait of interest. So some of the genes were in or neo calmodulin binding proteins, which have been shown to be involved in driving variation in bill morphology and Galapagos Darwin's finches. And also there were other genes that were involved in craniofacial development in humans and mice. So the interesting thing here is that even though there's different environmental drivers apparently for bill variation and channel song sparrows compared to the Darwin's finches, there's still some of the same genetic architecture that's causing this variation in the trait. So the last sub-question here is, are there loci associated with bill morphology? And she had 324 birds with both sequencing data and the bill dimensions that she then mapped to the zebrafinch genome, which is the best annotated bird genome. Fortunately, there's a lot of synteny with birds, so it's a pretty good genome for other species. Ended up with 11,000 SNPs to do this genome-wide association study. And what she found is that, once again, there's a lot of loci that are related to bill morphology. And also the loci she found related to bill morphology. So this is a Manhattan plot if you're not a genetics geek, just shows position on the chromosome, and this is the relative support that a given locus is related to whatever you're looking at, environment or the trait. And of the loci that were related to the trait, they explain a big whopping chunk of the variation. So what this means is this variation is not just due to plasticity. There's some genetic basis to a lot of the variation in the trait. And I was actually kind of surprised at this because RADseq, you're not looking at the entire genome. This isn't whole genome sequencing. So you're missing big chunks of the genome, yet you're still explaining a high proportion of the variance in the trait. And once again, when you do look at the gene annotations, a lot of the genes make sense. Once again, you have some involved in calmodulin binding pathways, some involved in morphogenetic protein signaling expression, which have been implicated in variation in bill morphology and Darwin's finches, great tits, etc. And also some involved in craniofacial development. So some of these make sense. I mean, you can always spin stories with when you do these sorts of studies, but a lot of them are exactly what we would anticipate. So when you look at all the different sub-questions and these different analyses, everything lines up suggesting that, yes, indeed, the birds appear to be locally adapted to climate. So going back to the original conservation question, when you're choosing the best source individuals from Santa Barbara Island, it means that you should probably choose individuals from an island that has a similar climate as Santa Barbara to make sure that the birds you're dumping out there are more or less locally adapted. So we're really hoping that the Park Service is going to pull the trigger and do this because it's going to be so fun to monitor what happens. So also stepping back to the general theoretical framework about the importance of interest-specific variation and determining vulnerability to climate change, what this shows is that there's a pattern of increasing sensitivity to temperature as you go from east to west, driven by climatic variation. And for bird species that can easily fly these distances, this represents a case of microgeographic adaptation. Now, of course, this isn't the whole picture for understanding vulnerability because you still have to understand what are the current temperatures, what are the projections and increase in exposure to these ions. So I'm not claiming that by doing this, we already know relative vulnerabilities, but it's an important first step. And next time you go see a bird on the street right after this talk and you see some pigeon, you start paying attention to its beak. remember that that's a radiator, it's not just a mouth. All right, so that's the main thing I want to talk about in terms of empirical results. But I also want to, I just can't resist telling you about some of the emerging results from our tailed frog work because I love tailed frogs. And once again, it's basically the same thing with trying to understand relative vulnerabilities to climate change. That clock is wrong. All right, I'll speed it up a little bit so we have a few minutes for questions. All right, so this is all part of the NSF Rules of Life grant. When you look at the RP and like, what the heck are they saying they want? It's really confusing, but somehow we fooled them. And basically the goal of the program and also the goal of our project is predicting vulnerability to climate change by linking environmental heterogeneity to genetic and phenotypic variation. It's basically the framework that I've already talked about. And the funnest thing about this project is I got to pull together some of my favorite scientists. So I managed to hire Britta Forrester, who's just an awesome landscape geneticist. After she finished her postdoc with me, she got a job with a species status assessment team that helps with listing decisions for the U.S. Fish and Wildlife Service. So she just started that job. On the physiology team, I had Amanda Chiquina, my PhD student, Alicia Shaw, who was my former PhD student, now assistant professor at Michigan State University's Keller Biological Station, Cameron Gallenborn. And the stream ecology team, some people might know this guy, he's a local resident, Jason Dunham. He works at the USGS Forest Rangeland Ecosystem Science Center. I work there, but I still don't know the name. And Leroy Popp is a well-known stream ecologist at CSU. And Aaron Langmuth from University of Montana. These are simulation modeling. Use the sort of the math brainiac behind it all. And so the framework for this grant is identifying spatial patterns of vulnerability to climate change and tailed frogs within and between species using this integrative approach. This was the conceptual framework we drew up to explain what we're doing. And then we're looking at all sorts of different layers of data. But without going through this in detail, we're just trying to understand the environmental template well, relating that to microevolutionary processes and phenotypic variation, focusing on critical thermal maximum temperatures, and then ultimately tying this all together with spatially explicit simulation modeling to identify the spatial patterns of vulnerability to climate change. So it's ambitious, crazy, we'll see if we pull it off. But the coolest thing is the amazing tailed frog. Why is this an amazing species? Well, it's amazing for a lot of reasons, but one reason it's amazing is that the tailed frog family, Escapidae, diverged from all other anurans, all other frogs, 200 million years ago, right? So if you look at opinion like a frog phylogeny, there's the tailed frog, and there's like everything else. So it's basically the coelacanth of the frog world. Another reason they're cool and why they have their name is that the males have this extended cloaca, which they use for internal fertilization, these fast-flowing streams. The only frog in the world that has this trait. Another thing that's cool about them is that the tadpoles have these sucker mouths that they use to clasp onto the rocks in these fast-flowing streams. I've heard of stories of like the fisheries biologists who go out in electroshock and they get tailed frogs. They take them, they stick them on their ear, make little earrings out of them. But the reason that we're really excited about them and why we're focusing on this for this grant, other than an excuse to finally work on tailed frogs, 20 years after I said I would, in my graduate research fellowship that Mike Bullen helped me write, which was going to be on tailed frogs, but ended up being on something totally different, was that they're really cold sensitive. And so, or warm sensitive, I should say. They live in these cold mountain streams are thought to be particularly sensitive to increasing temperatures, and that could be important in terms of ecosystem function because these tadpoles can be the dominant grazers in these streams. So there's two species, the coastal tailed frog here in Oregon, and then the interior Rocky Mountain tailed frog out in Montana. And so what I want to talk about now, I don't know why that came down, is there any evidence of local adaptation of sensitivity traits and or genomic data? So it's similar, analogous to the song sparrow, although we're not looking at bill dimensions, we're looking at critical thermal maximum. And we're just focusing on this part, we're not getting to the vulnerability part yet. So just quickly, here's the general study design for Montana. We did basically the same design at Oregon. But just for starters, we're doing 2 elevational transects, 1 low, 1 high elevation population there and there. Not a mass amount of difference in elevation, but fortunately, there's still a ton of variation in stream temperatures, about a 10-degree range in stream temperatures among these 4 streams over relatively small spatial scales. So importantly, this sets the stage for the potential for thermal adaptation to these streams. So then Amanda looked at critical thermal maximum, and what she found is that there's a lot of variation in the critical thermal maximum. Sometimes as much variation as you see among different species of amphibians. And also this variation perfectly matches the variation in temperatures in these streams. So you can spin an adaptive story. So now we're doing the genomic aspect of this project led by Grad Forrester. And here we're using whole genome sequencing. So we teamed up with a group at Berkeley. We made a really nice annotated reference genome for tailed frogs, one of the few reference genomes for amphibians out there. And with those 40 frogs from Montana, we ended up with 34 million SNPs, so quite a few more than just the RAD-seq approach. And a lot of these loci are related to CT max and temperature. So what this means is that CT max is genetically based, it's highly polygenic. It also means there's evidence for divergent selection among these populations driven by variation in stream temperatures. We're just in the early stages of looking at the function of these bazillions of genes. You have to go through this long process of deciding which genes you're going to focus on, because there's way too many to look at them all. So looking at the top of the top candidates, and even though we're in the early stages, some of the ones that you expect to see are popping up, like heat shock protein modifier. So already, this is suggestive that you can reject this sort of classic hypothesis that sensitivity traits don't vary across the temperature gradient. And so this, once again, suggests that we need to stop thinking of these species as fixed entities with fixed traits when we're doing these climate change assessments. It's exciting, but we still got a lot more work to do. We're going to genotype way more sites and individuals, and so we're going to use an approach called Untitled Chapter, where we zoom in on the loci of interest as well as a set of neutral loci, an additional 22 sites, 272 individuals, both in Oregon as well as Montana, using this hierarchical spatial sampling design. And then ultimately, we're going to try to pull it all together using the simulation modeling approach developed by Aaron Languth, this framework called CD Metapop. And ultimately, what you get is some sort of map of spatial variation and relative vulnerability. And this is what we said we're going to do in our proposal, but sometimes things happen in your proposal that you didn't say you were going to do, such as catastrophic wildfires and once in a millennium heat waves, which we know are no longer once in a millennium, right? And for better or worse, good for science, bad for people, this fire landed right on top of some of our study sites. And so 2 of our study sites were engulfed in this, the Holiday Farm fire that scorched 173,000 acres, burned all the towns along the McKenzie Highway, and almost torched H.J. Andrews Experimental Forest, where we do a lot of our work. But in terms of answering questions, the good thing is that some of the sites are in the fire, some are outside the fire. So these ones are affected by both the fire and the heat wave. These ones only by the heat wave. And so Amanda, through pure determination and stubbornness, managed to trudge through this massive burn, which is unpleasant environment to work. And just the preliminary data shows that in the heat wave only sites, temperatures increase a little bit. That's the red dots compared to the blue dots. But in the fire sites, temperature went through the roof. All right, and these are temperatures these are like the highest temperatures we've seen in any of these escapist streams. And then the preliminary CT max data, she found that in all the heat wave sites, there was a significant increase in CT max, a little bit. Three things happen in the fire sites. You're going to get the highest CT max you've ever seen. So for some reason, some mechanism, CT max has increased just like a little over a year after these fires. So it could just be acclimation. But what I'm not showing is that Amanda's done a ton of acclimation experience. We never see this kind of acclimation ability in those experiments. And so the question here is this rapid evolution? Because all the ones that don't have the high CT max just bite it and they just can't survive. And so what's left are the ones that have the wheels that allow them to tolerate these high temperatures. And so I'm happy to say we got NSF Rapid Grant that are specifically for these sorts of situations. The fun thing is that my former PhD student is now the PI, now the co-PI. And so we're going to use this integrative approach, like all my research, genomics, fieldwork, experiments, to try to dissect what are the mechanisms that are allowing this response, because this is important. This, you have to just accidentally have things set up when the catastrophe happens to do this. Oh, by the way, if any of you guys know any awesome undergraduates or postdocs, or you want to help Amanda this summer around here, around Andrews, email her, or you can talk to me later about it. I also just want to say that extreme events are no longer rare. So this is a different NSF Rapid Grant that we had funded 5 years ago, where we were studying extreme insects. The once in 500 year flood lands right on top of our study sites, and then we got rapid money to go back and look at community and genomic responses. So these are no longer real. Like I just this morning talking to the graduate students, like everyone's had extreme stuff happen to their critters. So we have to be prepared to test organelle responses. This is going to happen at all of our study sites. All right, so just finishing up really quickly here. So the two big questions I laid out were, first, how common is interspecific variation in sensory traits? Well, I've only looked at 2 out of 2 million organisms that have been described. But it suggests that it might be pretty common. And it's also somewhat predictable based on the environmental template. And also, this is happening at a really small geographic scale. So adaptation is happening at a finer scale than I think people recognize historically. So I think we need to stop thinking about traits as these fixed entities, and we have to pay attention to the variation, because that's super important for on-the-ground management, because the way managers work, they don't like say, oh, this species is more vulnerable than this species. They say, which populations can I work on? And so you need to help them identify what part of the range is most vulnerable. All right, so just some parting words. This is all super depressing stuff. Like, it's tough to be a biologist nowadays. So I just want to end with a few kind of words of encouragement and hope. So now we've had this, pandemic, climate change, and looming on the horizon, biodiversity collapse, and finally the general public is just starting to understand that this wave is probably even worse than this wave for human well-being. And I think it's best said by J.R. Tolkien when he wrote, "I wish it need not have happened in my time," said Frodo. "So do I," said Gandalf. "And so do all who live to see such times, but that is not for them to decide. All you have to do is decide what to do with the time that is given us." And so for whatever reason, you all were born in you're doing graduate school in probably the most important 20-year period on the history of humanity for determining basically life's survival on our planet. And that seems dramatic, but it's true, right? But now it's more important than ever that scientists are at the table. And so this is just a handful of some of my friends who are engaging in policy. Really hard work, super important. This is just a little story about how I've kind of taken this diet as well. I was invited to this working group in England, and it was like all these fancy evolutionary geneticists and little me. And we were the only ones that actually focused on the job, which was integrating genomics into policy. And it was tough because I had to learn how to communicate with conservation practitioners who speak a totally different language than we do. But now we're getting invited by U.S. Fish and Wildlife Service to help them figure out how to integrate genomics into listing decisions, for example. That's it. That clock needs to be fixed. Thank you.
I just want to say thank you to Dr. Funk, and I think we have a couple of minutes for questions. If there are any questions online as well.
The process part. That's me announcing with the questions on the chat, so no worries.
Yes.
How are you quantifying or I guess determining the critical thermal temperatures for tadpoles?
Yeah. So yeah, it varies among species that you do this. And I'm not a real physiologist, I work with physiologists. But essentially what we're doing with tadpoles is you have them in this chamber, experimental chamber. You're ramping the temperature up. I think it was 1 degree every 3 minutes, I think is the rate of ramping it up. Well, first of all, I should say you're activating for 3 days. You don't feed them because you don't want food in their gut to affect the metabolism, to affect the results. And then we got to do an experiment, you're slowly ramping it. And for these guys, since they're already plastered on a rock, you know, they could actually be completely dead and still be plastered on a rock. You wouldn't even know it. So what you do is you like squirt a little mini like pipette at them once in a while, like constantly basically. And the point at which they just like fall off the rock is CT max. So CT max is this finicky thing, and a lot of people are like, is that ecologically relevant? And that's a good question. But the cool thing is that Amanda did this experiment where she, it was a different kind of experiment where we're looking at growth rates, trying to look at T-opt, thermal optimum, as a function of what temperature these tadpoles are held in for different amounts of time. And she did from basically like 5 degrees to 25 degrees. And in the 25-degree treatment, some of the tadpoles, after like 3 days, just died. They couldn't handle 25 degrees. And even at 20 degrees, some of them died. So then she looked at the relation, that was unintentional. The point was to look at growth responses at different temperatures. But then she went back and looked at the relationship between CT max for the same populations and the proportion that died. It's like 80% of the variation is explained by CT max and the proportion that died. So it suggests that CT max actually is relevant to individual fitness in different temperatures and different populations. So the populations that had really high CT max, they didn't die in the 25-degree treatment. The ones that had low CT max with colder streams, they didn't do well at all. Yeah.
How did you determine the gene sets that you used that were associated with CT max?
How did you do what?
How did you determine the gene sets that you used that were associated with CT max?
Well, so initially what we're doing, so we did 2 passes. First, we did whole genome sequencing on 40 individuals with that sampling design I showed you, 2 elevational transects, one as low as we could go, one as high as we could go, so hopefully extreme temperatures along the gradient. And then sequence those using whole genome sequencing. And then at that point, we're looking across the entire genome. So we're looking at everything for that set of individuals. You know, there's some issues with probably power because we're only doing 10 individuals per site. And so that's, you know, that's not ideal power for a GWAS study, but there's this trade-off between looking at the whole genome and the number of individuals you can do. And so that first pass, we identified thousands of variants that relate to CT max. We also had a good annotation from the reference genome we had developed specifically for tailed frogs with our Daniel Roxar at UC Berkeley, who does this with amphibians for a living. And then the next step is with the MyBase, we're just targeting like 1,500 of the top candidates that are highly correlated to CT max or in genes that make sense, are also related to temperature, et cetera. And then we're going to look at variation in a lot more individuals at sites using that targeted capture panel so that then we can increase our sample size to see are these genes that are, or modifiers that are commonly associated with that trait or that environmental gradient across the landscape. Does that answer your question? Okay.
I'm going to ask some more about the diet and the song starts. So I study crossbills. I'm assuming you're
Cool. Yeah.
familiar with that system.
Yeah, totally.
And it struck me that the test that you used to say that these bill differences are definitely not diet would also, you'd also come to the same conclusion for crossbills. And then, you know, there's pretty strong evidence that the bill structure is related to the diet in those guys. And I was wondering more if you have like behavioral observations of what they're actually eating on the islands.
Yeah.
And if the cacti are part of that variability across those islands.
Yeah, that's a good one. That's a good question.
Yes.
Yes. Yeah, Maybelline. Yeah, first of all, we acknowledge that this is not the world's like the ultimate. Like, if you really want to conclusively cross out diet as playing any role, you'd have to do other things. So I recognize that. Maybelline did do some behavioral observations. She found that really hard to find what they're eating because the little finches are eating all sorts of little seeds. And she did a lot of that and kind of just abandoned it in the end. She has not done stable isotopes. So that would be a logical next step to really dive deeper and see if there is something different with diet that requires variation in build morphology. I wouldn't be surprised if there's some role of diet. Maybe it's like multiple selection, multiple environmental gradients that are driving this variation.
My other question for that is that it looked like, I don't know what temperatures you're showing, those are just averages, but that's still like the high 24 degrees or whatever, still pretty, within the thermological zone.
Yeah.
So like, does it frequently push them to like a point where you would expect?
Yeah, well, actually, those temperatures are kind of, it's crappy data. That environmental gradient is not, the real extreme temperatures are much higher
Yeah, I figured.
at a finer scale. But that was like the GIS data that Maybelline had available.
Yeah.
And she did do the experiments where she's pushed them to their upper thermal limits and lower thermal limits in boxes and doing like intensive physiology. She does get to temperatures where it's causing responses.
I just wondered if they were experiencing those on the island.
Yeah, that's another good question that I'll probably like send you to Maybelline to ask. She's like the real bird physiologist on the team.
Cool.
Yeah, that's the danger of
really
being an
cool.
integrative biologist because I can't be an expert in all this.
Yeah. Thanks.
Yeah, Mike.
That's another question, your opinion about reintroduction studies and choosing source populations. So you can study particular traits like you did, or you can just take the genomically most similar, or you can mix them all together, let selection sort it out, the genetically diverse population, or study particular traits like you did. A cynic might say, well, it's kind of obvious that just given no other information, just pick the ones from the most similar habitat.
Mhm.
For example, which island is going to be most similar to the target population? I'm just curious, is it going to be more like the eastern one?
Yeah, it's tricky because like in terms of probably average temperatures that might in some ways be true, but like Santa Barbara's this teeny island, so it's incredibly influenced by the marine climate. So it's got like more fog than Cruz does. So there may not be like a perfectly analogous island. But to your larger question, like, yeah, this is like a huge, interesting area of investigation. And but I don't know the answer. The problem is that you get one introduction and there's all these approaches to do it. And so you can't do like some experiment to try them all and see which one works the best. I think probably what we're leaning, and I think in a lot of cases you're not gonna have all this data to help you make your decision. And you're gonna have to do something that's like seems like common sense. Like let's use birds that come from a similar climate as the target island, or we have museum specimens of the Santa Barbara ones. We can use ones with similar build dimensions, right, without even any genomic data. Or you can go the approach of just dumping in individuals from different populations and letting natural selection do the work. The island populations are not so divergent that you should be worried about outbreeding depression. And we know that they actually do breed naturally sometimes when they fly from one to the other. But at the same time, I think it's when you have opportunity, it's nice to actually have another 4 layers of data to have more confidence. When you make these big decisions and Park Service is gonna like dump birds on this island, and there's a lot of resistance and environmental impact statements, they want as much data as they can get to say, you should be thinking about adaptation because we have some decent evidence that there is local adaptation when you make this decision, and you should probably do the best you can to choose some birds that are likely locally adapted to the island. So long story is, I don't know the right answer. Hopefully we're going to do it. We're going to monitor it and see how it goes, and we'll see which individuals persist, have higher fitness, and don't, because the plan would be to do like a full pedigree of all the birds on the island to look at things like you do with steel track.
Another question, actually, just about the birds. You actually had an enormous rate of just observed dispersal among your islands. How does that jive with FST? Is there selection against migrants?
Well, we always saw the structure plot. We saw
Migrants, yeah.
one individual from crews go to Miguel and one go to Rosa. And we're looking at a total of each of those islands have, like Miguel, we had over 100 birds, so it's 1% of the birds were migrants. And with Rosa, it was like 0.5% were migrants. So what do you call it, like a huge amount of dispersal? Is there selection? I mean, theoretically, if there is adaptation and you have a higher fitness with the bill size of your natal island, there should be some selection against those individuals that migrate because they won't have the optimal bill size and maybe other traits, other physiological traits for that environment. So I don't know if there's, I mean, you could do the modeling, right? And you could figure out exactly how strong selection would have to be.
Yeah, that's my question, because it's a huge rate of gene flow. So I was wondering if you guys had done that.
Yeah, with the large NEs, it ends up being, relatively high rate. So we haven't done that. That's a good idea to potentially measure selection. I mean, probably some people want to go.
I was
I'm
going to
happy
say,
take
I
your
think we
questions.
need to slowly wrap up, but you can go ahead and talk to Dr. Funk one-on-one if you'd like to. And then he'll be at the graduate student lunch, and there is a happy hour as well for grads at 5 o'clock at Fitness.
Thank you. Question?
Sure. I did actually good. Sorry.
Yeah.
Yeah.